So today I would like to explain certain features of general recombination, and in that we will see introduction, initiation, homologous pairing, branch migration and resolution. These two aspects follow the Holliday junction model, and in the end I will be explaining you the Holliday junction model of general recombination. So just let us see what is general recombination and what are the requirements for a general recombination to take place in prokaryotic or eukaryotic cells. Now, general recombination is a process of DNA exchange between homologous chromosomes. This particular terminology is very important. You have to understand the term homologous recombination. When we say homologous sequences, we mean the exact match of the nitrogen bases on the two DNA sequences. If the exact match is achieved, what the difference remains is, as we have already discussed, the methylation of the bases. DNA, they are on the nitrogen bases methylated at different sequences, different nitrogen bases, even if there is a complete 100% homology, and that is what it makes the exogenote and the endogenote differ from each other, even if they are similar in their nitrogen bases. But before going to that, I would like to just explain some of the features of general recombination with respect to the requirements. Certain enzymes and proteins are required. The first thing which is required is a Chi site. So as I was mentioning, one of the first requirement for general recombination in a cell to occur is the site on the DNA on which the enzymes can load. They can come, they can get attached and start the process. One such sequence on the DNA is called as the Chi site, and it has a very particular arrangement of nitrogen bases from 5' end to 3' end. It is very specific. As I read it out, remember it: GCT, GGT, GG, from 5' end to 3' end. This Chi sequence is present on the DNA, one strand of the DNA, and this is called as an octamer because 8 nitrogen bases form this Chi site. This is one of the most repeated sequences on the DNA for during recombination, and therefore it is called as the most overrepresented octamer in the E. coli genome. Why this is present repeatedly? Because the attachment of the proteins which initiate the recombination, that is a random event, and translocation is a process by which movement of these enzymes takes place on the DNA. During translocation, it will just come and attach at a site where it can start the recombination process, and that site is called as Chi site. One more important activity of this Chi site, as you can see written there, is that it modifies the nuclease activities of RecBCD proteins. These RecBCD, which are important for recombination to initiate the recombination, they nevertheless have one more maintenance function when recombination is not taking place, and that is degrading the DNA. If there are free 5' ends and 3' ends, remember this is endonuclease type of enzyme which goes on degrading the free DNA. So this is the nuclease activity which is modified by the Chi site and helps in initiating the recombination. So you can attribute two functions to Chi site, that is first binding, second modification of the RecBCD's nuclease activity. The next immediate requirement, as you can see, is RecBCD. It is a multi-subunit enzyme, a protein of course, and it has two prime functions, helicase and endonuclease. RecBCD, it initiates the general recombination by attaching to the blunt ends of the double-stranded break in the DNA. I have explained in the last lecture what are blunt ends, and actually though cohesive ends are more useful in the recombination process, and that is what we use when we are artificially using this process in recombinant DNA technology. But in natural process, this RecBCD initiates general recombination by attaching to the blunt ends of the double-stranded break in the DNA. Using ATP molecule, means it requires energy, it moves along the DNA, that is called as translocation. It translocates along the DNA strand, unwinding the strand. Therefore it has a helicase activity. It unwinds the DNA, and the rate, as we have seen earlier, that it is 1000 base pairs per second at given temperature of 37 degrees centigrade if it is observed in the laboratory. This endonuclease activity is asymmetric across the strands, and it is modified when the chi sequence is encountered. I will show this in animation, that it has a nuclease activity, that is, it goes on breaking the DNA till the chi sequence is reached. Once the chi sequence is reached, what happens is that the chi sequence, it modifies its nuclease activity and it loses its nuclease activity. Okay, so RecBCD also has a second function, that is, apart from being endonuclease, it has a helicase function, and it also is involved in the, what you can say, attachment of the Rec-A, its counterpart. It is counter subunit. All these subunits, multi subunits, that is Rec-A, Rec-B, Rec-C and Rec-C and Rec-D, they are required in combination. That is, a combo activity is there. But as we have seen here, RecBCD is a 3 subunit 1 molecule. It is a multi subunit enzyme molecule. The next second molecule needed is a single unit Rec-A. So once RecBCD comes, degrades the DNA, reaches the chi site, there Rec-A is free to join, and Rec-A is joined with the help of RecBCD proteins. So we have, you can say, 3 functions: helicase function, endonuclease function, and the third function is facilitating the attachment of Rec-A. Now we will see what is Rec-A. Rec-A is a protein which polymerizes, means it is a polymerizing protein. Its function is to provide stability to the single-stranded DNA, match the homology, means homologous sequences. As I said, endogenome is double-stranded, exogenome may be single-stranded. So you have 3 strands which should be in homology, means the sequence of nitrogen bases in all these 3 should be same. Again and again a question may arise in your brain that if all the 3 sequences are ditto, similar in their nitrogen base pair arrangements, then why is there a necessity for recombination? I have already explained this with respect to the methylation process. So you always have to remember that even though if there is 100% base sequence homology, the strands, they will differ in their function due to difference in the methylation or the methyl group on individual nitrogen bases. And the last function of this Rec-A protein is strand invasion, that is, invades the strand. So Rec-A, it forms a polymer, it forms polymers on the single stranded DNA, and it outcompetes the single stranded binding protein. Means in the cell there are single stranded binding proteins which will come immediately if a single stranded DNA is encountered, but these Rec-A proteins will outcompete, means they will move aside the single stranded binding proteins and they will function in their place when there is a general recombination taking place on, and this takes place eventually on the chi containing DNA region, chi containing strand. Okay, and of course RecBCD protein helps through this process. Now when the homologous target duplex DNA is encountered, when it is present, the Rec-A protein, it aligns the single stranded DNA with its homologous region on the duplex DNA, means you have 3 strands. I will show this in the animation and you can see that when I will end explaining all these enzymes. So 3 strands are there, the homology matching takes place, and through the triple stranded DNA intermediate, Rec-A, then it starts to replace the, what you can say, recessive gene with the dominant gene. Even if they are same, as I said, methyl group, that is methylation, determines which is dominant and which is recessive, and Rec-A then inserts the single stranded DNA into the target DNA and it replaces one of the pre-existing strands and it forms Holliday structures. I will also explain what is a Holliday junction model. The remaining three enzymes you already know. These are DNA polymerases, ligases. I will explain that in brief because we have already seen their functions in DNA replication, and the same functions are here. Genetic recombination follows a replicative path, means for that short segment where the exchange is taking place, there the replication is going on. So DNA polymerase is an enzyme complex and its function is synthesis of DNA strand. DNA synthesis during general recombination, this has to be remembered. So DNA polymerase, it extends a DNA strand or a RNA primer in 5' end to 3' end. The direction is very specific. During this process, as we have seen in replication, during this process, the RNA primer in the 5' to 3' end direction, means there has to be a RNA primer during replication, and here the existing DNA, which is the third strand, it will be extended, means there is extension of the DNA strand up to some basis complementary to the template strand. Okay, RNA primer in DNA replication. Here the exogenome itself is available for extension. Therefore, it plays a very integral part in replicating the DNA wherever and whenever it is needed. So here during general recombination, we will attribute the DNA polymerase function to extending the exogenome. The next is the very popular pasting enzyme which you know very well, ligase, whose function time and again we have seen in detail. So I will be very short here to tell that this is used in repair. It seals the short one-stranded DNA break which are there in the double-stranded broken DNA. The two nitrogen bases, the introduction of phosphate group. Such breaks may arise from the action of DNA polymerases itself, and when it extends a DNA strand, it doesn't join the DNA strand. Joining is done by DNA ligase. That is introduction of phosphate groups between two nitrogen bases. Now, this is one more protein which is called as Rhubarb protein. It is a tetrameric protein and it is the main protein which recognizes the Holliday junction. Okay, Rhubarb protein, it recognizes and binds to the center of the Holliday junction. When these three strands, you see three strands have to be converted into four strands, and that is where this DNA polymerase got into action, and it helps. Once the 2 plus 2 4 strands are there and they form a certain interlocking junction, you will see in animation that junction has to be resolved, and that is resolved by Rhubarb. Rhubarb recognizes and binds to the center of the Holliday junction model. It unfolds it into a square planar configuration which induces binding by Rhubarb. Now, having said all this, I will now immediately go into animation where whatever I have discussed, you can just experience the whole thing in motion. Bring your imagination into animation and see whether that matches or not. Okay, the next immediate protein is Rhubarb protein. Rhubarb. It is a ring-like hexameric protein and its function is as a molecular pump. Two Rhubarb hexamers, they bind on the opposite sides of the Rhubarb. Means Rhubarb is there from one side and one side to Rhubarb, and then from the opposite side. As I said, hexameric it is, and each binding is powered by ATP. ATP is required. You will see this in the animation. If you feel hard for understanding, don't worry, it will be shown in the animation. And each Rhubarb ring, it rotates the duplex DNA, passing through the central hole like a screw turning the nut. And what this does, this pulls the DNA through the Rhubarb tetramer, and with the result of branch migration, the generating regions will become heteroduplexes. A heteroduplex region will be generated, and then there will be a horizontal cut or a vertical cut generating two types of recombinant DNA molecules. The last type of protein is the Rhubarb class itself, and that is the Rhubarb protein. It is an enzyme, it is endonuclease. See, the Rec BCD proteins endonuclease activity was lost, so in the end you need some endonucleolytic enzyme which will carry out the process of cleavage. So two molecules of Rhubarb, they resolve the Holliday junction model, and by binding to Rhubarb, they can introduce cuts horizontally or vertically at opposite ends and cleaving and giving rise to two different types of Holliday junction model products. I will show you that. But let us see now the general process of recombination in animation. If you see, as I said, Rec BCD binds to the free blunt end of the double-stranded DNA. As you can see here, this is the one which is there. Then it moves along, and as you can see, one of the strand, it is getting degraded here. Degradation takes place, and degradation of the enzyme is then, once it reaches the Chi site, it is lost. Now Rec A, it mediates a homology search through the formation of triple strand, and as you can see here, what has happened is there is a, let's go back behind a bit. You see what is happening, homology search, and now what has happened, I have paused it. As you can see, GATAT is not even reached. Now I'll just play it and see. There is a sequence homology. Let's go back again a bit, and you can see here what has happened is, you can see CTATATATAGC, this GGGCCC. There is a sequence homology, and this sequence homology which is achieved, that results into the exchange. Now Rec A catalyzes the exchange of the incoming strand, and now as I said, polymerase, it extends the strand. There is a gap here. This gap will be filled by ligase, and now a double Holliday junction model, as you can see here in the figure, this is one and this is one. A double Holliday junction model results. Ruv A and Ruv B, they facilitate the branch migration, and you can see Ruv C endonuclease, it resolves the Holliday junction, and it results into two types of DNA molecules. There are two different types of DNA molecules. One, it will be called as a patched DNA molecule here in this animation, and the other is a spliced DNA. But to be more specific, I will take you to another animation which will guide you in detail about how the Holliday junction model takes place. So come, let's see the Holliday model. As you can see here, two duplex DNA, this region, they form a crossing over, and then they undergo a branch migration, forming a heteroduplex. The both heteroduplex, they bend away from each other at 90 degrees angle. Now remember, once there is 90 degree, the lower half of this structure undergoes a 180 degrees to uncross the crossover. Holliday junction is then cleaved either horizontally or vertically. Okay, horizontal cleavage, it gives to non-recombinant heteroduplex products, whereas vertical cleavage, it gives rise to recombinant products. The products are hybrid in nature. So as you can see here, there are two byproducts when a Holliday junction model is encountered. So it can be summarized in few steps, where in the first initial stage there is a branch migration, crossing over, and branch migration takes place simultaneously. Once the branch migration takes place, you have to remember there are two types of rotations of the strand. One is 90 degrees in the initial stage, and then uncrossing over results by 180 degrees towards the left, 90 degrees towards the right, and 180 degrees towards the left, due to which there is a junction model, a Holliday junction, it results. And when this happens, there is a cut introduced in the DNA, and that cut, as you can see here, this cut can be from the top to bottom or left to right or right to left. That is, you can say as a vertical cleavage or a horizontal cleavage, and this results into two types of products, a horizontal cleavage products and a vertical cleavage product. Horizontal cleavage products, it gives rise to a non-recombinant DNA. As you can see here in this particular figure, this gives rise to a non-recombinant DNA product. As you can see, A, C, B, it is non-homologous. But here, if you see in the vertical cleavage product, it gives rise to a recombinant DNA, where a segment of the trait is achieved. So this is the Holliday junction model. Hope you have understood it. You can just rewind this video, pause it wherever necessary, and take down the notes. Remember, videos are meant for repeated viewing, pausing and taking down the notes. So hope it helps for you to understand in the general features of recombination. Thank you.